Welcome to NSTA Engage Spring 21. My name is Heather Ryan and I'm the sessions moderator. Chris Herman and Rachel Mosey are supporting us behind the scenes today, and Chris Chow will be presenting Making Connections Between DNA and Chromosomes. Please use the chat during the session, and that chat will become part of the recording that is posted on the Spring Engage site tomorrow. During the session, Rachel will post links to resources, registration for your free student modeling pack, and the session evaluation. I will also monitor your chat for questions throughout the session. And we will let you know when you can unmute and interact. I now have the privilege of introducing Chris Chow. Chris holds a bachelor in bioengineering and a master's in curriculum and instruction from the University of Illinois at Urbana-Champaign. She's been a science educator for 20 years and enjoys facilitating workshops for science teachers in her district at national conferences and now virtual sessions. She received the 2014 Spotlight Award for Excellence in Teaching from the St. Vrain Valley Education Foundation and the 2016 Outstanding Biology Teacher Award in Colorado from NABT. She currently teaches AP Biology, Biotechnology, and coordinates the Medical and Bioscience Academy program at Longmont High School in Colorado. And now let's begin the program. Take it away, Chris.
Good afternoon, everybody. Thanks for joining in. I especially appreciate those teachers who might be sneaking in PD on the side while in between classes, or that you've taken the day off of work. So I feel like this is really valuable opportunity for teachers. So thank you for doing that. All right, so today in our workshop, we are going to cover chromosome structure and anatomy. We're going to take a look at mitosis and meiosis and how to model that using these faux models. Look at ways that these mechanisms of division of DNA can contribute to genetic variation, and then also connect DNA with Punnett squares and inheritance of traits. So what we're trying to connect is DNA at the molecular level to chromosomes more at the macro level. So in my classrooms, these pictures represent activities that I do with my students to incorporate modeling as I teach AP biology in particular. I also teach biotech and forensic science, so I try to bring in these models whenever I can. Of course, these photos are pre-COVID photos. You can see I do a lot of group work in my classroom and use a variety of the 3D molecular design kits. Models are great for showing how processes work within the cell. Students are able to do hands-on work with them, and it really reveals some of their ideas and also misconceptions. And so this chromosome kit is really important to reveal some of those misconceptions they have about cell division. So you as participants hopefully have signed up and received the student modeling kit. What you're seeing here is the classroom version in that photo right there. In my classroom, I usually have students work in groups of 4, and they have big spaces with those tables to be able to model the chromosome movement. And but for you as participants, and what I use this year with my students with a lot of virtual teaching, especially in the fall, were the student modeling packs. My students are so excited to get these and take them home. And so we're going to be using that throughout this workshop today. So you could go ahead and get those out. There's a little bit of assembly required. There's some cardboard pieces that you need to trim out using some Elmer's glue, glue on some of those foam pieces you see on that diagram, and then assembling the Punnett square graphic. If you don't have the chance to do it right now, no worries, or don't have the kit, they're super easy to do and easy to follow with, okay? So we're gonna start off with just having you use the kit right off. So here's a picture showing you a couple of chromosomes represented at a couple of different levels. So go ahead and assemble that, and I'll give you a little bit of time here. The important thing is you don't necessarily have to do the identical version that you're seeing there, but the fact that you have the larger chromosomes that you're pulling out and assembling, connecting them to these graphics, and then to getting some of those nucleotides on there. The sequence of the nucleotides is not important right there. Okay, and as you assemble that, I'm going to turn on my video so that you can see my overhead camera there. And my picture, my little camera will show and I'll model along with you as we're doing this here. So I'm going to leave my camera on and you can see that little model right there. And we'll move on to the next slide there. So as you and your students assemble this graphic, it was intentionally built this way to be able to show representations of eukaryotic chromosomes at several different levels. So what I would ask you to do is to see if you can identify from these levels what you see in your model itself. So at the top, you see DNA represented in the double helix format with the base pairing of the nucleotides, that twisted ladder that students are used to seeing. From there, you have DNA organized around the histone proteins. And then as there's further wrapping and condensing of the chromosomes, you have nucleosomes coiled into a chromatin fiber, and then further condensation. And at the very bottom is showing you the traditional picture of a duplicated chromosome when chromosomes have already been replicated and are shown in that way. So as students see this organization of eukaryotic chromosomes at different levels, you can have students compare how does your model represent these different levels of organization. And you can have students hopefully recognize from the molecular level of the nucleotides and then the organization of DNA within a cell all the way to the coiled chromosome format. So this is a great graphic to show that transition because students often see the nucleotides, they see the chromosomes, but they don't really understand how conceptually they get from one to another. So this middle graphic is really great for connecting the two. All right, now we're going to, I'm going to have you see that in this picture that that's on this slide, the student modeling pack is on the right with the nucleotides in the red, green, yellow, blue colors for each of the different bases. And in the classroom version, you have the color bases, but also the, actually what you have are the red nucleotides and blue nucleotides to represent maternal and paternal chromosomes. So you could still do the base pairing there, but you don't have the 4 colors shown. So the other thing is, I believe this kit also has gray nucleotides to show parent strand and daughter strands if you want to show the differences there with replication. Okay, let's go ahead and now go to sort of the larger scale, looking at coiled chromosomes here. And you want to assemble the chromosomes shown. Again, the number of linkers that you're seeing on that screen is not super important, but just showing that for ease of demoing this, the processes we're going to take a look at of having maybe 2 larger chromosomes or longer chromosomes and 2 shorter chromosomes. So I've got that showing on my overhead cam. You will have to remove the chromosomes parts off the ends of your original graphic that we were taking a look at just a moment ago so that you can assemble these chromosomes right here. Okay, so a red and blue of the longer chromosome and a red and blue of the shorter one. In that middle section, I have them, one of them offset and one of them kind of in the center. You can arrange them however you want to show 2 different lengths and maybe 2 different locations of where those center areas are. Okay, so let's take a look at what all of these parts are demonstrating. So the ends that are kind of round are the telomeres or telomeres, the ends of your chromosomes. And you could see the blues represent paternal chromosomes and red represents maternal chromosomes, the ones you inherit from mother. And the center area, and the blue and red with the 2 short guys and the 2 longer chromosomes, those are homologous chromosomes where they have the same genes and number of genes in relatively same length and location of that centromere that's in the middle, which is kind of pinched in there. Those are the homologous chromosomes. So you have 2 shorter pair of homologous chromosomes and then a 2 longer pair of homologous chromosomes. So this is a great way for students to get a few terminologies, like few terms straight in their head, because I think in terms of cell division and with chromosomes, there's a lot of C words. There's chromosome, there's centromere, there's chromatid, and having students point and identify things is really helpful. Okay, so as we take a look at what these models represent, each one of these foam long pieces represents chromosomes. And for our purposes, within a human cell, this is a human karyogram showing you all the chromosomes organized by length and the staining pattern and the location of the centromere. So we have 23 different types of chromosomes. Of course, the last one are the sex chromosomes, X and Y. And then within a human cell, we are diploid creatures. So we have 2 copies of every chromosome. So you can see these homologous pairs represented in this ideogram. So when you transition from this graphic, you want to also show students a karyotype, a picture of a person's chromosomes aligned in that order to see the staining pattern, the sizes, location of centromeres, and potentially any abnormalities in terms of number of chromosomes or pieces or sizes of chromosomes. And of course, you can see this individual with 2 X chromosomes biologically is their sex is female. Okay, so another connection to make with students is thinking about how many chromosomes there are along the cell cycle. And of course, this graphic shows that G1, S, G2, and M phases of the cell cycle, and that cells doing their everyday normal thing is in the G1 phase, and that DNA gets replicated in the S phase of the cell cycle. So DNA synthesis and DNA replication happens there. And that's important to point out because by the time you get to the M phase, that's where chromosomes are, where the cells are grown in a petri dish and the photograph is taken of the karyotype. So that traditional karyotype that you see is those chromosomes already having been duplicated or replicated because students don't often realize that when they see that graphic. So that's an important point to make note of for your students, where replication happens and that picture of when it's taken. Okay, so we're going to do that S phase and replicate your chromosomes. So you're going to take the additional foam pieces that you have from your kit. You're going to replicate every set of chromosomes. So I'm going to do that right here. Mine are pre-assembled, so it's going to take me a lot less time than perhaps you doing this kit for the first time. So you can remind your students of the machinery involved in this replication, which is, you have your helicase, DNA polymerase, and all the other enzymes involved. And that's at the molecular level, so part of our replication happening there. And so you would not normally see these chromosomes in its replicated state, nicely coiled and showing in this format until you actually get to prophase of mitosis, where the chromosomes can start to condense up. So that's important to point out as well. But for our sake, one of the things I want to highlight is for students, I like to have them hold out one of these pieces of chromosomes and say, or one of these molecules and say, this is a chromosome, and then this is a chromosome replicated, to indicate that as a molecule, this in this state is still attached, is one chromosome, and this is a chromosome, but this is a chromosome replicated. So using that language is very important to understanding when we come to cell division and students are asked to identify how many chromosomes are in a cell, that they recognize this is actually a chromosome, and when they separate during anaphase, that's when you say there are 2 separate ones. Okay. All right, so each of these chromosomes is made up of 2 identical sister chromatids. So holding this out, this is a chromosome replicated, and each one of these pieces or segments is a sister chromatid, and they are identical. So Again, with all the C terms, chromosome, chromatid, centromere, students get confused quite a bit. And so this kit will help you to have them point to it, talk to each other, and be, for me, actually, I have them get up physically and say, like, I'm a chromosome replicated, and put out their arms and legs and be like this kind of X formation or replicated chromosome. So a lot of hands-on and body movements in my class. All right, so let's take a look at what happens after we replicate our DNA. And so, of course, the cell in doing mitosis is aiming to make clones and ensure that there's an exact number of chromosomes get into each one of those daughter cells, the identical genetic set there. And so you can take your foam models now of your chromosomes and demonstrate. Hopefully you have some space on your table there or around you, maybe on your floor, to show the different stages of mitosis from prophase. Now these chromosomes become visible. And I want to show a couple of things. So when my students were at home, they were at home, and so these student modeling kits come with these foam chromosomes, but they didn't come with spindle fibers. So you could just have them take yarn or string or whatever you want, pipe cleaners, and model those spindle fibers and kind of lasso them. I kind of do that, and then show the movement of chromosomes, with the alignment of these chromosomes attachment to the spindle fibers there. In the classroom kit, you actually get the kinetochore right here and the spindle fiber. So you can attach it to the centromere right there. So you can demonstrate this in the classroom, and students can use the classroom kit to do that. So that's super handy. All right, so let's take a look at, I think there's a video showing that animation of, all right, and then we have duplication of the chromosomes, and then separation of the sister chromatids so that you end up with two identical copies of the parent cell in the two daughter cells. So you can ask your students these questions, and to show you visibly with these models, daughter cells compared to parent cells, the number of chromosomes here. In the student modeling kit, you have a total of 4 chromosomes. In the classroom kit, you have enough materials to assemble and make 6 chromosomes, so you can have 3 pairs of homologous chromosomes there, and to be able to ask these questions in terms of keeping track of the math of how many chromatids or chromosomes in each cell. So let's kick it up a notch here and go to meiosis. Cell division for the purpose of making gametes or sex cells, sperm and egg. And so you could see here's the graphic. What's nice about this in our kit is you have blue representing paternal, red representing maternal chromosomes, and where all these chromosomes are going. So we're going to go ahead and model these parts of meiosis. Okay, so going through the cell cycle, we have G1 phase, and I'm going to show this on my overhead cam here. G1 phase, we have each of our individual chromosomes, not coiled up, but eventually they will be. And then S phase of the cell cycle, we have replication. So here we make identical copies of our chromosomes, okay? And next comes, we have, of course, G2 phase, they're already duplicated. And then we have the next part, which is crossing over. So this schematic shows how crossing over is actually a more complicated process of where one strand of the sister chromatid invades the other sister, where we have the homologous chromosomes pairing up or forming a tetrad here in synapsis. And you have the one sister chromatid and the other sister chromatid, and they actually have strand invasion. So there's gonna be pairing and a breaking of the chromosomes, and this crossing over that happens at this location where they join together called a holiday junction. And the purpose, what's shocking, the purpose is not necessarily genetic variation, but the purpose of this whole process is to ensure that these homologous pairs of chromosomes are lined up and attached to each other before, as they're getting lined up in metaphase 1, alignment at the equator. So this is a point where which mutations can happen, and hopefully it's done accurately. And if there's a point at which that there's some breakages, hopefully there's some DNA repair happening, but it is possible to pass on some of these mutations in the mistakes that happen with crossing over into daughter cells. So let's show this process with the classroom kit. It's kind of the fancy version here. You can see the strand invasion, that's the holiday junction, that repair happening, and now you have crossing over. You can see sort of the mixing of the DNA pieces of maternal and paternal chromosomes. All right, so that's showing at the molecular level, and now we want to show at the more of the larger level of chromosomes, visible structures. So you can do two ways. You can show how you can have these longer homologous chromosomes there, and show a crossing over where you have one piece breaking off at the same location on the homolog and just swapping the pieces like this. Okay, and you can show that. All right, and I'll talk about my little label here in just a second. Or you can do what is in 3D space. You can actually put your chromosomes on top of each other because we have the capability of being in 3D space. So you can see they're stacked on top of each other, and you can show in 3D space how you can have the breakage, alignment and breakage of the chromosomes and the swapping of the pieces in 3D space like this. So you can see that crossing over happened like that. This is kind of great to have this model that students can handle because we're not showing crossing over in a 2D flat format, but you're showing it in a 3D space so that when if you're showing what's happening with the chromosomes and they separate out, you could see that if I line them all up in 2D space, you could see that's how that crossing over happened. So in an actual human chromosomes, there's 1 to 3 chromosome crossing over events per chromosome. Now, I do want to do a shout out for all our AP Bio teachers out there. AP Bio teachers, I'm going to flip over my chromosomes here. And one of the concepts we teach in AP Biology is the fact that there are different genes on the same chromosome and they can be linked. So I've got some traits here: body color, wing type, and eye color here. Of course, those are our famous like Drosophila flies and their traits that they can inherit. And students have a hard time understanding this recombination that happens when chromosomes are close together versus far apart, and that recombination frequency is not as often if those genes are close together versus genes far apart on the same chromosome. So, and then you can illustrate that in this way, and it's really helpful for students to see that and why that recombination frequency number changes and what happens if these genes are on different chromosomes. Okay, so little tip for AP Biology there. So at home, you could take your replicated chromosomes replicated, and start to move them to show meiosis 1 through prophase 1, metaphase 1, anaphase 1, telophase 1, and then meiosis 2. So you can see when you count the number of chromosomes, you start off diploid in prophase 1 and end up haploid in each of those cells in telophase 1. So you only have, instead of 4 chromosomes, you're now down to 2 in each daughter cell. And then the second division, you start haploid, and then you're splitting those sister chromatids. So you still end up with 2 chromosomes in each cell. So I can watch the graphic there, the animation. All right. Okay, so you can see in there, the animation, you have the yellow kinetochores and microtubules or spindle fibers shown there. So that's the classroom kit. I like to, in my classroom, with the tables that I have, use the highlighter markers, not highlighter, but those expo markers that are really bright, the window markers, and have students draw the nuclear envelope or the cell membrane to show where that would form or disappear, and be able to show on a table where those, what's happening there. So that's a little bit easier for students to do that in a classroom. So the other thing with that animation is that was a stop motion animation. Chris or Heather, could you guys play that one more time again? And so this was made by 3D but your students could totally do this themselves as well with the stop motion app that's on iPads or phones. And so this is a great way for students to demonstrate their learning using these models as well. So little caveat to that, it looks like the video goes for, 10, 15 seconds. And of course, as everyone knows, anything done digitally takes 50 times as long. So you might want to plan like half a class period for that. But I think there's great learning opportunities for students when you have them model that and video it and explain what's going on themselves. So questions to ask your students with meiosis. How did the daughter cells compare to the parent cells? Do the chromosome counting, how many are in each stage? At the end of meiosis 1, at the end of meiosis 2, and then maybe have two different groups of students compare how are mitosis and meiosis similar and how are they different. Those are important pieces to be able to elicit student understanding and ask those questions. Okay, so thinking back to the ways in which DNA was being shuffled or mixed up, what are the sources of genetic variation for offspring? And so you can ask your students that to realize it happened during crossing over, just like we see here in my camera here, happened when you're aligning those homologous pairs of chromosomes at the equator during metaphase 1, and then the separation of them in anaphase 1. And then of course, sperm and egg only have one copy of each type of chromosome. And so you could have a lot of different variation in the sperm and egg genetic content to combine during fertilization. So a lot of different places to point that out for students. Okay, so one thing that would be helpful using this kit is to be able to bring in a case study or some kind of application with all of this once they understand making connections between the DNA level to the chromosomes. So I'm going to have you guys play the role of genetic counselors. And so we're going to meet a couple who are the Smiths, and they're wanting to start a family, but they know there's a history in their family of a genetic condition called sickle cell anemia. And so when we take a look back at that, let's bring it to the DNA level. So the gene involved is the HBB gene, or the beta globin HBB, which codes for the beta globin protein. And the hemoglobin molecules, which carry oxygen, have 4 of these subunits, 2 alpha globin molecules and 2 beta globin subunits. And within them, there's that heme group to carry oxygen with that iron in there. And so we're taking a look at just that beta chain, or the beta subunit of the hemoglobin protein itself. So where is this gene? Let's put this in context. It's located on chromosome 11. And when we zoom into chromosome 11, it's located on the P arm there at that location. And when you zoom in even closer into the gene sequence, for one particular variant of the HBB gene mutation at a particular location, and this is where you can take your foam nucleotides now and create these different variations. So at a certain location, you can get a couple different variants. So here I've constructed these. Now this is an actual what you're going to see here. And the reason we have 3 instead of just the difference that you see there is that one spot in the middle is because this forms a codon with that triplet. And then you can connect it to that codon will eventually transcribe into mRNA, and that gets translated. So you can see at that middle location, instead of having a T down at the bottom there that you're seeing that it's highlighted in yellow, you get a point mutation where you have a substitution of an A. So an adenine instead of a thymine. And when that transcribes to RNA, you get GAG in a normal beta globin gene, and you get the amino acid glutamic acid. That is a hydrophilic amino acid side chain, and we know that those chemical properties are really important in the folding of proteins. Now, if you get an adenine instead of the thymine, it transcribes into GUG, which when a ribosome reads that, will code for a valine. And that is a hydrophobic R chain or side chain. And so let's take a look at what will happen if you get valine instead of glutamic acid. So in normal hemoglobin, that glutamic acid is when that globin, a beta globin molecule folds up, it is facing the outside. The watery environment, it's hydrophilic. So those side chains on the exterior part of beta globin are hydrophilic. But when you substitute that amino acid for a valine, that valine is hydrophobic. And what that creates is these hydrophobic pockets and creates sort of a sticky spot right there where it's attracted to other abnormal hemoglobin molecules with that hydrophobic part sticking out. And as these molecules kind of clump together, it forms this big long chain, as you can see on the right side. Okay, so let's connect it back into when we have Punnett squares and look at genotypes and phenotypes. Genotypes is showing what's in the genes, right? So you can have students model with their foam nucleotides. What would it look like for 2 copies of the normal hemoglobin? What would it look like for 1 copy of each or 2 copies of that abnormal sequence there? And so relating it to genotype then with the big H for the normal variants, or big H, little h for being someone who's a carrier, or little h, you are affected with sickle cell. Turns out, if you have big H, little h, you don't have the sickle cell disease, but you do have what we call the sickle cell trait. For the most part, these individuals, we call them carriers, but they do experience some physical difficulties or symptoms in different conditions. So for example, at high altitudes, where that load of, carrying oxygen is kind of a challenge there. I live at 5,000 feet above sea level. So definitely people who come here from sea level, like my folks visiting, experience some breathlessness and shortness of breath. And so that also happens with individuals with sickle cell trait. They might be just fine at sea level, but experience some respiratory issues at high altitudes, for example. And so you could talk to students about how will the genotype and the structure of these hemoglobin molecules affect its function and its ability to carry oxygen in red blood cells. It's a good connection there. So taking a look at the cell level, what would we see under the microscope? Normal hemoglobin molecules would be nice and round and globular, and consequently your red blood cells are nice and circular, roundy shaped. And then sickled RBCs or red blood cells are that sickle shape because all of those molecules kind of sticking together, forming these rod-like structures, will stretch out the cell membrane and the structure of it. So it makes it more difficult for those cells in the next graphic to show cells going through capillaries, which are very narrow. Normally red blood cells go in single file line. So if you have sickle-shaped cells, you're going to end up having more blood clots and issues in different organs. So back to the couple, Mr. and Mrs. Smith. You do genetic testing on them and find that they're both carriers. They have sickle cell trait, and they want to know what are the chances if they have offspring of their child having sickle cell anemia. So you can now use your graphic here. I'm going to put that under my camera here. So you can have your graphic out, and this is where you get to show Punnett scores. And instead of having just big H, little h, you can help students to understand individuals are inheriting the actual genetic material from their parents. And so you can have them model these variants at these loci on their DNA. And you can show that, let me make sure I'm orienting this correctly here, that these individuals have, right there, okay, so one of each variant of this location of the sickle cell gene, of the HBB gene, and then show how different combinations of these chromosomes, which have these variants, can be inherited by their offspring. So as you click that graphic, you can see, have students move down for each of these possible boxes to show possible combinations of these alleles. And if you're doing this digitally, you can have, or remotely, you can have students take pictures of each possibility and maybe put in a digital file to record that. There won't be enough nucleotides in the kit to form all of those alleles shown, but you can have students move down and show different combinations of these alleles in each of these boxes. Okay. All right, so as the big question is, what are the chances that the Smiths will have a child affected with sickle cell disease? Genetic counselor will walk through them with how that comes about, what that means for their child as carriers, that their child will have their children have a 25% chance of inheriting sickle cell disease, and you know, to guide them on you know, some of the resources to be able to help couples who are facing that. Now, of course, this can also connect with CRISPR, because there have been a couple of quite a few cases in the news now of showing how CRISPR can be used to help individuals who are affected with sickle cell disease in reverting one of the genes, not the HBB gene, but turning on a different version of hemoglobin in their cells to be able to help them be able to carry oxygen better. So anyways, CRISPR connections there, really great for talking to students about that. So thank you so much for attending this session. I hope you and your students are able to find a lot of different uses for this kit, whether they are virtual or in person. And I'm happy to stick around for a few minutes here to answer any questions you might have.
Great. Thank you, Chris, for a wonderful session. And please go ahead and post any questions that you have in chat. And this is also a time when you could unmute and turn on your camera and ask questions directly of Chris. So while we are waiting for things, I'll go ahead and we'll do our closing. So we wanted to remind you that Chris was showing you some of the student modeling pack, the chromosome student modeling pack today, and the chromosome connections kit. So please make sure you filled out that registration to be able to get your free student modeling pack. And then you can use the code 3DMD Engage 21 for 20% off most of our products now through the end of June.